To see the study you're conducting as a kind of recipe you use to make your food, you write down for somebody to um, cook the meal when you're not there. Cook the meal when you're not there. So it is very important that this aspect needs to be transparently done. Needs to be transparently done. Um, so that is the key thing here when you're conducting the study. And then at the analysis stage as well, you need to, so those that have not muted, I'm meeting you. So if you're not speaking, then you get yourself muted.
Yes,
So
I just wanted to make a request.
yeah,
I said, is it possible to give us a sample of the protocol, how
yeah,
it should be?
I can include it in the Dropbox. I will open a folder, protocols,
Okay, sir. Okay,
and
sir.
include in the Dropbox. So if you want that, that is of course the 1001
Thank you so much.
protocols there. So I will include it in the
Thank you.
Dropbox.
All right, sir. Thank you.
Yeah. So you develop the protocol before you start the study, and when you come to the analysis stage, you should also prepare your protocol, your analysis protocol, because when you do data analysis, it's not a straightforward thing. You need to think about it carefully. You need to think about how the raw data you have collected, perhaps you need to transform your variables. You need to clean the data. You need to make different um, editing to the data. And once you have done that, you need to also think about what method do I use for my analysis? What method is appropriate? For those who are used to fitting models, how do I develop my models, and so on and so forth. So that needs to be thought carefully. And this will usually depend on the question you are answering, the question you are trying to deal with. So the protocol for your analysis needs to be developed, carefully thought. And once that is done, you start the analysis. When you clean the data, the codes you use, the syntaxes you use to clean the data, all need to be intelligibly, as we discussed yesterday, described, intelligibly described, and make it transparent. Actually, like I showed you one of the protocols we did last time, you can actually publish your protocol, but minimum you can register it in the platforms that register these protocols. And so the data, if the data is possible to be made public, you should also include the data so that somebody can take your codes and try to reproduce what you have done and see that you have done it rightly. So that is one um, initiative at the analysis stage, and there are other initiatives we mentioned. This is when you're doing the analysis. The I showed you how you can make your analysis plan and so on. And then this is initiative at the publication or dissemination stage, in which different types of reporting guidelines have been developed for um, for people to use to guide the reporting of their papers, their manuscript, and the goal here is to ensure that your papers, your manuscripts are reported in a transparent manner. So if you go to Equator, you see this. And then we went for lunch and we looked at peer review. the peer review, and we looked at conventional versus open peer review. And there is a movement has been going from the conventional towards open peer review, um, all with intention to make what we do as scientists transparent and reproducible. Um, and so this is one initiative as well at the dissemination stage. Yeah, Cecilia is here. She will come in, but let me finish this up. And so we mentioned these other initiatives that are there, and we talked about the impact factor and so on, incentives, which was the great debate we had. So these are some examples of the initiatives, and you can choose from one of these and elaborate, develop your own perspective, or you can actually work on your own what you think within the context of Africa, what you think can help to increase reproducibility. So that was my thoughts for that project. You can select one of these, elaborate each, or you can develop your own considering your context, what would be useful in that situation. Okay, I hope it's clear for you.
Yes,
Yeah, I
sir.
see, Cecilia, you're welcome this morning. Sorry, perhaps we didn't have everything settled in terms of communication, but yeah, you are here. Olayinka, it seems you have a question. Please go ahead, and after you, will allow Cecilia to walk us through.
Okay, thank you very much. When you asked whether there is any question so far on what you have discussed, that was when I raised up my hand.
Yeah.
Now, yes, when we talked about registration or publishing of protocols before the actual work. So now I wanted to ask, now, like in my field, biochemistry, when we are running the project, we say so parameter or so thing was determined according to the method of and you can describe that method a little. So the published protocols we are talking about, or the protocols that can be raised, are they novel protocols that you developed yourself, or all these protocols that have been developed in time past, say for so thing, I will use so method, for so one, I will use so method. So that's what I just want to get cleared, sir.
Well, a protocol is not really equivalent to a method. You can use a protocol to describe a method. So you may have a standard method that is used for certain things, measurement, and so on and so forth. But the protocol usually is a document that describes what you intend to do. So the protocol can be novel to the extent that you're describing your study.
Okay.
It may not be novel to the extent that similar study has been done before. But even if the similar study has been done before, let's say you're doing a study in the Democratic Republic of Congo,
Good. Yeah.
and somebody has done a study, for instance, looking at anti-malaria
Yes. Yes.
medications, and the other
Yes.
person wants to repeat such study in Egypt. In
Okay.
Egypt, a person
Yes.
develops a protocol. One is that there are differences between those studies. The first one is that the subjects that have been included in those studies are not the same.
Yes.
The investigators are not the same.
Okay.
And you may have other things that are different across, between those studies. So in that sense, they are different. And so the protocol you develop is novel to the extent that it is not the same, exactly the same people you are studying or the same thing you're working with. And so that is why we encourage that every study needs its own protocol. Okay.
Thank. Yeah. Thank you.
Hmm.
No,
Okay,
but like
let's.
someone requested, we appreciate if you can have maybe an already published protocol, maybe
I
one or
will.
two.
I
Just
will do that. I will do that.
I really appreciate it. God bless you, sir. Take care.
I will do that. Okay. I'm based here in Sweden, so you can see Cecilia is a Swedish, and she's a colleague of mine. We are both working at the same center, which is called Crafting Research Center, which is based at the Institute of Medicine, University of Gothenburg. And what we do there is a very dynamic center. We are really proud of the center. It's not a big center as you used to know, but we are growing really. And so we are working primarily on issues of allergy, respiratory disease, and so on and so forth. And then our center has research from very little molecules to big human beings, giants as well. So we cover everybody as far as it is related to health, biology, or so on and so forth. And we also have a big lab and people who are also doing animal studies and so on. So we have epidemiologists, those who are doing clinical epidemiology, population epidemiology, and so we have coverage of the translational spectrum of research. And so I am on the other spectrum of dealing with human beings that you can touch, you can see. Cecilia is on the other spectrum where she's looking only at the molecules. And so she's dealing with this very tiny molecule. She will explain that. I don't know how those work, but it's quite fascinating what she's doing. And so she has been a very good colleague. And in relation to this course, when I teach it, she's always coming to bring aspect of reproducibility in her research. So I've invited her this morning to take us through what she does, how she does it in her field, and what we can learn from that. So Cecilia, over to you.
Thank you, Bright, for that kind introduction, and my apologies that I'm a little bit late. I had 9:30 in my calendar, so I was sitting with other things. But let me share my screen.
Sorry about that.
Oh, no worries. Maybe it was my fault. Okay, so I'm hoping that you're seeing my screen now. So
yeah, we're seeing that.
Perfect, thank you. So today I'm going to talk about implementing reproducibility in basic research. And as Bright said, my name is Cecilia Lesser and I'm at the University of Gothenburg here in Sweden. And I thought I would introduce myself a little bit. So who am I? I'm a molecular biologist. I got my PhD in 2013. And I'm working as a researcher. And as Bright said, we're really working with the smallest molecules. We're culturing different cell lines. We take blood samples and other airway samples from the patients. But in the end, we're analyzing the proteins, RNA, DNA, and lipids of these samples. And I'm specifically working with extracellular vesicles. Maybe some of you have heard of them before, but they are these vesicles that cells release to their environment. And there are a bunch of different ones. So we have this smallest one, the exosomes, that are formed through the multivesicular body. So it has interluminal vesicles, and when it fuses with the plasma membrane, these are released. We also have the microvesicles. They're a little bit larger. They are budding off from the plasma membrane, but also dying cells will shed off apoptotic bodies. And together we call these extracellular vesicles. So it should come a picture here, but maybe it's a little bit heavy. Here it is. So these are the vesicles when we look in cryo-electron microscopy, because we cannot see them with normal microscopes. We have to go to electron microscopy. So here you can see 200 nanometer in size, and we have a bunch of these beautiful vesicles of a little bit of different sizes. And vesicles are released from, here you can see bacteria, where that vesicles are budding off, and then you have them all the way to mammals, and so really it's a very conserved way for cells to communicate with each other by releasing these vesicles. And they're everywhere. So if we look in the body, we find them in blood, saliva, urine, and so on. They're really breast milk. They're everywhere. We also have them in the environment, so sea, water, soil. And since yeast and bacteria is releasing them, we also have them in fermented food. So if you're having beer or wine or other fermented food, you're actually eating these vesicles. And as I said, they have a lipid membrane. In this lipid membrane, we will have some membrane proteins, some adhesion molecules, some tetraspanins. We also have a lot of proteins on the inside. We have RNA on the inside, so the RNA is protected by the lipid membrane. So when we realized that these vesicles contain RNA, it was really a big finding, because it means that cells can send microRNA and mRNA between each other, and this RNA will not be degraded because it's protected. And it's also suggested that these vesicles contain DNA. So why are we interested in these vesicles? Well, they participate in physiological functions, so coagulation. We see that pregnant women have more of these vesicles in the circulation that are released from during the whole pregnancy. But the big thing, I think, where a lot of research is done is on the functions in the pathological conditions. So, for example, tumor-derived vesicles can suppress the immune responses to escape the immune system. It can promote pre-metastatic niche, and they can facilitate angiogenesis. So this is a big field, but here we are also interested in the vesicles during airway diseases. And then the last thing is that since cells release them and they contain RNA and proteins and so on, they are a good biomarker candidate because they represent the deceased cells that release them. We are also interested in loading them with different molecules because they are constantly circulating in our body, so they are well tolerated. And if we can manipulate them and engineer them to contain different drugs, we can use them for that as well. So I'm usually thinking of them as little message in a bottle that are floating around in our body. And they participate in all this different kind of cell-to-cell communication. And when I started in the field in 2007, it was less than 100 papers per year that was published. So I could basically read them all. And nowadays we have over 2,500 papers per year. So it really is an expanding field because we've seen that these vesicles are part of so many different pathological processes and the biomarker and all these other fascinating things that these vesicles can do. So that was a little bit of background about me and my field. And of course, you're not interested in the vesicles necessarily, but I thought it was good to give you that because I want to talk about how we have tackled the problems with reproducibility within our field. So as I said, this is a rapid expanding field. Thousands of new papers coming each year. It means that it's a lot of new people entering the field, which is super exciting. They're bringing a lot of new knowledge, but it also leads up to the problem that not necessarily everybody, when you're coming into a new field, understand the complexity of the field. So you have to understand that you need to use the right controls, because for this particular fields, we have problems that you don't have if you work with cells, for example. These vesicles are so small. It means that there are limitations to the techniques used. So these are nano-sized and not micro-sized. So we have to think about these as well. And of course, everybody have a favorite method from the people who have been in the field a long time. And then we have this new exciting people joining the field that maybe even developing new methods. And because they have new skills and they're coming maybe from the biotechnology field and so on. And this kind of have to merge to understand how the new techniques are fitting with the old techniques. So data don't lie. If I go down in the lab and I do an experiment and protein A is expressed, for example, in a Western blot, I mean, the data don't lie, the protein is there. So if another paper have suggested that protein A was not expressed and I find it, so, you know, if I can't repeat what somebody else have published, there's a couple of explanations for that. And the first one is, of course, the most problematic one, that somebody have committed fraud. But we also have failed transparency, so that the people who published haven't been transparent enough for you to repeat it in a good way. And these two is what the researcher that performed the data the first time could have done. These two bottom part, which is poor experimental design, execution, and analysis, but also carelessness in the use of reagents, materials, and method and statistics, can actually be both by the researcher that performed it the first time, but also by you who are repeating it. Have you repeated it in the same way as the other group did? I'm going to take an example of fraud that was actually in my field. So this is an EM picture of vesicles, and you can see there's a lot of nice vesicles, cup shaped, which means that they have probably have a double lipid membrane, so they have inflated a little bit during the fixation. This looks really great. It was published in 2014. The problem was just that another paper was published in 2005, and you can actually see that this yellow vesicles here is the yellow vesicles here, while the green one here have been cut out from here, and so on. So it really is that somebody have photoshopped this picture so that instead of actually doing an analysis of their own sample. And of course, this paper, when this was acknowledged, was retracted. So why do people commit fraud? Well, science is a competitive environment, and people are pressured to obtain results. They need that to publish. They need it to get grants. They need it to get a tenure or get promoted. And there's also a big desire to publish in high-profile journals. And it has actually been shown. So here we have impact factor for different journals. And we also have retraction index. And you can see that many of these papers with high impact factor also retract a lot of papers. So what is it by the higher impact factor journals then? Well, this study asked people what factors contributed to this irreproducible research that can be the ground for retracting a paper. And I thought it was interesting that one of the top one is selecting reporting. And I think that is a problem for many of the high impact paper journals, that they want a clear message. They don't want, well, we saw this, but we also saw that. And I think then people are kind of picking. They're picking the results that are supporting this clear view, while they're not lying, they're not changing any figures, they just leave maybe some results out that doesn't fit the story as well. And of course, if somebody's repeating it and focusing on this part, the story is probably much more complex. But you also have a lot of other things: pressure to publish, or you have low statistical power or poor analysis. So you haven't analyzed the data in a correct way, or you haven't replicated it enough. Of course, fraud is here as well, but I think most of these things are more innocent mistakes, so to say. Okay, so data don't lie, and we have talked about the first thing, which is the fraud. Now I'm going to talk about failed transparency, because I think this is really where you and I can contribute to whichever field we're in. So as I said, my field of extracellular vesicles is expanding really, really fast. So in 2014, a paper was published which highlighted the minimal experiment requirements for definition of extracellular vesicles, so MISO. And they propose a series of criteria based on the current best practice that represent the minimal characterization of EVs that should be reported by investigators. So basically they said, if you want to say that you have isolated extracellular vesicles, if you want to claim that they're doing something, this is the minimal experiments you have to do. And this was published by the board of the society. But in 2018, an updated version was published, so MISO 2018, and actually everybody in the field who wanted to contribute was invited to do so. So I think this paper have close to 280 authors, and basically a set of questions were asked. So do you think this is important? Do you agree that we should demand that people do this or that? And it was really a survey of what do the researcher in the field think is important if you publish within this field. So I'm just gonna talk through some of the points that were raised in this paper. And the number one is there is no single optimal isolation method. So this is a problem. There's a many different ways that you can isolate extracellular vesicles. But you have to choose it based on the downstream application and science questions. And this is just to show several different methods to isolate and the studies that have used it. So it really is a lot. So what they are requiring then is that, okay, we're not going to tell you that you have to use this method to say that you have vesicles. But what you have to do is that you have to describe in detail the method that you actually have used to isolate your vesicles, because that will allow interpretation and replication by other researchers. But we can only do that if you are specific about what you have done. So this is an example. We use ultracentrifugation to pellet the vesicles. As I said, they are very small, and we have to do 100,000 G to pellet them, compared to cells that you can maybe pellet at 300 G. So this is usually what was written in the methods section maybe 10 years ago. A ultracentrifugation step at 100,000 G for 70 minutes was performed. Okay, so we can start with the first problem. Because the G-force is a calculation where you have the radius within this calculation, which means that you can either calculate that you have 100,000 G in different parts of the tube. So if we start with a type 70 rotor, which have these different radius at the minimum, average, and maximum part of the tube, we can see that if we calculate 100,000 G at the maximum radius, it means that we will run our samples on approximately 31,000 RPM, because we have to calculate the G forces and the RPM, because RPM is what we actually put into the instrument. But if we instead would use this calculation and say, yes, we want to have 100,000 G, but we want to have it at the average radius, then we should actually have almost 37,000 RPM. And if we calculate 100,000 G here and we run with this RPM, then we actually have 140,000 G at the maximum. And of course, this can sound like small things, but really, if we're all doing different things and cannot repeat what other people are doing, this is really important if you don't understand how these things are connected. So another thing is also that we have several different types of rotors. So we have the fixed angle rotor, which I showed earlier, where the tube is still during the centrifugation and is usually then have a short migration path. And then you have the swinging bucket rotor. And when you start the centrifugation, it will stand up from the rotor and out. So you actually have a longer migration path. And we compared two of these rotors. So we started with the TI-70 rotor, and we isolate vesicles and we measure RNA in our vesicles as the yield. And then we took a swinging bucket rotor and had the exactly same G-force and time. And then we got less yield, we got less vesicles. So it was the exact same G-forces and time, but our yield was lower. And then we realized that, or I mean, we knew this, and this was what we wanted to highlight, that the swinging bucket rotor has a different K factor, so it's not as good in pelleting, and you actually have to centrifuge for much longer time. So there's actually a calculation you can make. So we use the calculation. So these are two different rotors. They have different properties, but the web page for the company has a web page where you can calculate the differences. And when we got this and we run it much longer, now the yield were the same. So of course, if you're using this rotor and you get less yield than your colleagues or what other people have done, this could be an explanation. So it's important to know that the rotor has an impact on what kind of vesicles you isolate and how much. And therefore you need to describe which rotor you have used in your method section of your paper and where you calculated the G-forces in this rotor. And as I said, this is from Beckman-Coulter because we're using their ultracentrifuge. Here you can very easily say, I have a type 70 rotor. Now I want to change to a type 45 rotor. I want to run for this time on this speed. You press calculate, and then this calculation tells you, okay, 70 minutes with this rotor is like 94 minutes of this rotor. And that's what we've shown in this paper as well, that we now published a few years ago, that these rotors are not, you cannot just take one and take the same G-forces and the same time, because it's huge different. For example, this rotor is super efficient. So 70 minutes with this type 70 rotor, that is very common, 27 minutes is enough here. But for some other rotors, you actually have to go up to almost 2 hours. So it's very, very different on which rotor you use. And this is just to highlight that as well, that here you have the diameter of the vesicle, the size of the vesicles, and here you have the percentage that you're pelleting at a certain time. So here they have done 30 minutes. And you can see that while this rotor pellet 76% of the 100 nanosized vesicles, if these are the vesicles you're interested, this rotor only pelleting 14%. If you calculate them, as I said, you can use this K-factor rule and on the webpage of the company, these move much closer to each other, you can see. But it means that you have to centrifuge for longer time for the rotor that is not as good in pelleting. So in 5 years, we went from something like this. An ultracentrifugation step at 100,000 G for 70 minutes was performed to obtain exosomes. And now instead we say exosomes was collected at 100,000 G average. So it has been calculated in the middle of the tube, a type 45 rotor with this K factor from Beckman-Coulter was used for 70 minutes. This means that other people, if they have a different rotors, can make the calculation themselves, how long time they would have to centrifuge to get an equivalent efficiency in the pelleting. So a few years ago, Anne Hendricks, a researcher in Belgium, started EV-Track. So EV-Track aims to increase transparency of reporting in EV research. So basically, if you have a paper, you will get a score on how transparent you have been in your method section. How easy would it be for somebody to repeat what you have done? So EV-Track, it's an open source knowledge base containing methodologically parameters of extracellular vesicles related publications. It's guided mainly by the MISIV guidelines from 2014 and 2018, since the field have said that these are the things that we think are important for people to report. And the EV-Track data is then mining to identify missing controls or inaccurate reporting for experimental parameters. And in the end, you get this score on how well did you perform and report your experiments. So you can see it as a checklist. Have I reported all these things? Because sometimes it's easy to forget. But for somebody else to be able to report and reproduce what you, or interpret it and reproduce what you have done, this information is crucial. So why was EV-Track established? Well, what I have highlighted before as well, you have this expanding rapidly field, and it's very complex due to heterogeneity. We have several different methods, and we need a systematically centralizing experimental parameters to provide transparency for reviewers, editors, and readers. So basically in EV-Track, you have to fill in this information about sample, isolation, and characterization of the vesicles. And then in the end, you get a score. And to be fair to other people, I have picked one of my own papers. So this is a paper that was published a few years ago. And as you can see, we got a score of 78. And that, I guess it's fairly good, but of course you want 100%. And you can easily see here, okay, so what were our problem with this paper? Well, it says that we haven't specified our antibodies. So this is actually what we wrote in the method section of this paper. For the Western blot, the membrane was incubated with anti-calnexin from Santa Cruz. So we thought that, you know, we had been very specific. It's the dilution, the company, and everything. But then if you go into Santa Cruz webpage and search for calnexin, you actually had 14 calnexin antibodies. So of course, if people would like to use the same antibody, this information is not enough. You need to specifically also say which clone or actually the catalog number. So this is a paper we published last year. And here we got 100% because, of course, we realized that these are things that we need to report much, much better. So one of the things that are suggested in the minimal experimental requirements, the MISIV guidelines, is that you need to characterize the vesicles so that you're sure that you actually have vesicles, because as I said, they are so small that we can only see them with electron microscopy, and we have to prove that we have vesicles and not just soluble proteins, cell debris, and so on. So the guidelines suggest that you have to report the amount of several proteins, both EV enriched and non-EV enriched. It's not enough to show that you have vesicles. You have to also show that you don't have other things that can be responsible for the effect that you're seeing. So they suggest that we look for transmembrane proteins to prove that we have vesicles with a membrane. We should also show that we have cytosolic proteins, so that we have intact vesicles that are protecting these cytosolic proteins. But there are also proteins that should not be there, that are coming from different organelles, such as Golgi and ER and mitochondria and the nucleus. We should show that we don't have this, to show that we don't have a lot of cell death in our cultures, and we just have cells breaking down, and this is what we're pelleting. So in this paper, it's very nice illustrated that they have firstly looked at CD63. It's a transmembrane protein. They show the presence. They then look at 4 different cytosolic proteins. They showed the presence. And then in the end, they have 5 different proteins for different organelles to show that these are not present. So when I'm reading this paper, I'm very confident that these authors have vesicles in their preparation, and that they have done a good job in determining that. So this was actually one of the problems we had in our paper from a few years back. We didn't get a score because we hadn't looked at non-EV enriched proteins. And I would say, in my defense, this is one of the things that a lot of people don't report. So EV-enriched proteins, the majority of the papers in the field are looking at these, but very few are then looking at the non-EV-enriched proteins. But of course, we have improved since then. So, and I'm going to show you now why this is important to also test for non-EV markers and report the contaminants. So here we have isolated vesicles from plasma. Plasma is a very complex body fluid, and we use size exclusion chromatography. So we load our samples on the top, we let them go through a column, we collect fractions in the bottom, and what we could see, so flotulin is a EV marker. We had a lot of flotulin in fraction around 7 to 10. So we were really happy, and we're like, okay, you know, we have vesicles in these fractions. These are the fractions we should focus on. But then we looked at ApoA1, which is on lipoprotein particles. So we don't want to have this in our sample. And we had massive amounts of this. So we could see that, you know, yes, we have vesicles, but we also have a lot of other things in our sample. And when we looked at the EM picture, I mean, we don't really see any vesicles because the sample is so dirty. So what does this mean? Well, we have our vesicles, and here we have the size. So they are maybe between 50 to 200 nanometers. Then we have the density, and the vesicles would float on a density between 1.1 and 1.2 gram per milliliter. So when we have used its size exclusion chromatography that's separating based on size, we can remove everything to the left of the blue line. That means we have our vesicles here, and we can remove the HDL, the LDL, IDL, and small VLDL because they're all smaller than the cutoff for the column. However, the size of the chylomicrons are the same as our vesicles, so these will not be separated if we only separate on size. But as you can see, they have a really, really low density. So if we add a density step, we can remove these, separate these two as well. So that's what we did. We loaded our plasma samples first on a density gradient or a density cushion. Then we took the samples that are in this density and loaded on a size exclusion chromatography and separated on size. And now you can see that we have a lot of flotillin, so a lot of EV markers, and we have almost been able to eliminate the contaminating lipoprotein particles, so upway. And now when we look at the EM pictures, you can see that we have our vesicles here and we have a much cleaner background than this. So for some studies, this is crucial to also look what other things you have to really understand your sample. So why is it so important to also test and report for contaminants, and why are we talking so much about these things, reproducibility and reporting, all the things? Well, you have this paper here, which I took away the authors and the journal. It's not important who it is, but in EV-Track here, they get 0 score. They didn't look at EV-enriched proteins. They didn't look at, they didn't specify antibodies, probably because they didn't use any antibodies then. They didn't score green on a single of these parameters. But they're claiming that exosomes derived from human bone marrow mesenchymal cells promote tumor growth in vivo. That's a pretty bold statement. These vesicles are promoting tumor growth in vivo, yet they have not in one single step showed that they actually have vesicles or provided enough information for somebody else to repeat or interpret the data. So this is another thing that is in this minimal experiment requirements. It is that you should characterize single vesicle by at least 2 techniques. So for example, you can look with electron microscopy. So you actually see the vesicles, or you can use a nanoparticle tracking analysis, where you can measure the concentration and the size and see that the size is within the size of vesicles. And it's also saying that if you're doing TEM, so transmission electron microscopy, the image should show a wide field encompassing multiple vesicles, in addition to close-up images of single vesicles. And I'm going to explain why that is. So for example, these are all my own unpublished images as well, so not putting anybody else to shame. But let's say I want to prove that I have vesicles that are approximately 50 to 100 nanometer in size. You know, I would publish this and say, okay, look at these perfect 2 vesicles here, proving my point. I have isolated vesicles of this size. But what I actually have done is that I have cut the image. And here I have a very big vesicles, and maybe, you know, I don't want to show that because I don't want it to be there, but it is, and you have to be honest with that. And that's why you need to show a big image that contains several different vesicles, so the reader can make an interpretation of what you actually have isolated. And this is another example. This is a vesicles, and the little black dots is golden particles, and the golden particles bind to an antibody. So in this case, I would use this picture and say, look, my vesicles are CD63 positive. And I mean, this looks great. But if you show the full picture, you actually have a lot of vesicles that are not CD63 positive. And you also have some staining that are actually unspecific, so it's not even with a vesicle. This is why you need to show the full picture and be honest about your whole sample. And then just some final things from the minimal experimental requirements is that if you do functional studies, you should do dose response. You need to process controls. to rule out the influence of serum components. For example, if we have a cell culture, we will have FBS, fetal bovine serum. We need to look at all these things. And we also need to do density gradients to show that the activity is in sync to the EVs and not just associated with soluble proteins. So, I mean, I hope I convinced you that we will all benefit from being more transparent and report these things. So then the question is, why do researchers fail to be transparent? So as I said earlier, many times journals often seem to prefer simple, uncomplicated stories. You also have word restrictions, and often you do want to have your discussion and your results. So the word restriction, you're cutting your method. So the method section gets shorter and shorter, and you take away more and more information to be able to fit this word restriction. It's also that there might be, or what this paper found when they asked researcher, is that you're actually not saying exactly what you're doing, because then other people cannot repeat exactly what you're doing. So that makes you a little bit unique and ahead for publications, positions, and grants. So you actually don't want somebody else to be able to do exactly what you have done. So why is reproducibility so important? Well, science progresses by findings from one researcher being advanced by another. And that's what I think is so cool with the research field, that a group in USA can have a finding, and I can build on that in Sweden, and then my finding can be the foundation for somebody else. But then we're depending on that what people are publishing is correct. Also, drug development builds on preclinical studies. And if we're gonna, if we have published a lot of things that are not fraud, but they're necessarily not correct because you didn't have enough statistical powers and things like that, that takes a lot of time, money, and energy if we're then gonna screen for drug development. So although we are competing for the same grants, the same positions, the same publications, we are performing this research to cure and help patients, and that should be our focus. So can we inspire researchers to be more transparent? Well, the EV track suggests so. In 2012, so this is before the MICEV guidelines in 2014, and it was before the EV track. On average, the score for papers were 21%. 2019, the average score was 54%. And it was also shown that here you have paper published before MICEV and paper published after MICEV, and you can see that the score has gone up a bit. You can also see that papers citing the MICEV guidelines usually had a higher score than papers not citing the MICEV guideline. And I think you see this specifically on these non-EV enriched proteins, that paper not citing the MICEV guidelines usually don't do this, while the papers that are aware of this are better on providing this information. So the final 2 points here. So we've talked about the fraud, we talked about the failed transparency. Then what's left is the poor experimental design and carelessness using reagents and so on. And this was, in this paper, they talked about poor productivity of biomarker research and development. So you have a large number of publications claiming putative biomarkers. You see them every week. Protein A, microRNA B, or whatever is a biomarker for disease C. But very, very, very few, a small number, actually enter a clinical validation trials that achieve final regulatory approval and clinical adoption. Why is this? Well, the paper then suggests that authors or researchers don't use fit for purpose biospecimens from well phenotyped sources. You have a small study, so you're lacking the statistical power. You have poor compliance on reporting standards by scientific journals. And then they're highlighting insufficient control of pre-analytic parameters. And this is the point I want to talk a little bit extra about. So, for example, if you're a person like me, you might be the person who go down to the freezer and pick up a plasma sample or something. And then from here, you start your experiments. But a lot have happened to your samples before. So you need to be aware of these things. You need to talk to the people who have done this, and preferably you should have been involved in this. So which time of the day was the sample collected? What do we know about the person? Did they have coffee, alcohol? Are they smoking? Are they on any medication? What happened during the collection? How was it processed? Was it transported? How long did this take from collection to storage? Was it hours, days? This is just an example. If you use blood, you have several different, to start with, anticoagulants. Then how did you centrifuge to get your plasma and serum? How long was it standing before that happened? How was it transported? How was it stored? All of these things are pre-analytic factors. These are pre-isolation. So for the EV field, this is before we start isolating the vesicles. These are post-isolation. So we have actually isolated the vesicles, but we haven't started analyzing them yet. But if I send my sample for microarray for looking at RNA, or proteomics mass spec to look at the proteins, these things might matter. So you really need to think about all the steps that are happening to your sample and report them, so you have them, but also report them in the publication. So be methodologically curious, be curious, ask other people. If you're not the one collecting the sample, talk to the people that are, and don't take things for granted. So for example, there was, if you're working with cell lines, it was shown that 30% of the head and neck cancer cell lines were misidentified when they were tested. Replicates are performed within an experiment, but it's not proof of reproducibility. Proof of reproducibility is if somebody else in your group can get the same result as you. Or can you get the same result, but from patient samples collected by another group? Or can another group get the same results as you? And so make new buffers, run several samples to understand what's unique for your sample. Don't, make just one Western blot once and it worked. I mean, repeat it to be sure. So in summary, to increase reproducibility in basic research, write the details in the method section when you publish. And if there is a word restriction, you can always add the details to the supplementary, where you can have a much bigger and more detailed method section. Share your method details with other researchers, because if a colleague can repeat your results, you have tested internal reproducibility. If a researcher from another lab can repeat, you have tested external reproducibility. And this is only strengthening your findings. This is good for you. This is meaning that what you're finding is true. Understand the method that you're using. What are the limitations of the techniques, so you don't draw conclusions that cannot be drawn? How does change settings affect the results? And how do I interpret and analyze the data? Pay attention to and understand the whole chain of your samples are going through, not just your part. talk to the other people that are either collecting the sample, or if you are the one collecting the sample, talk to the people that are going to analyze them. What are important for them? Because if you don't know what's important, maybe you're doing something to the sample. So it's really crucial to talk to each other within a project. And this is just a paper, how to make more published data true, which I just want to recommend. And then I just want to show that one of the last conferences or workshops I could go to before the COVID-19 situation was a very nice meeting that I went to in Belgium, in Ghent. And it was a workshop on open, reproducibility, and standardized EV research. And the chair was Anne Hendricks that I talked about earlier. She developed the EV track. We also have Clotilde and Kenneth, who was the organizer of the MySev 2018 guidelines. This was a really nice meeting discussing really important things. And I understand that for some people this sounds boring, but it is the foundation of our field. We are not stronger than this foundation and how we address these questions. So with that, I would like to say thank you for your attention, and feel free to ask any questions if you have thoughts.
Thanks very much, Cecilia. This is really fantastic, and it demonstrates the consciousness people are really having to improve what we do. And I think this consciousness is really established from the fact that even though you are dealing with very small, these small molecules, they have impact on human beings, because the goal is to improve our lives. And so if things are done in the wrong way, then the impact can be catastrophic to that extent. So thanks very much to come here and showcase what is happening in your field. And I think this is one example that every field has to learn. Of course, you can't copy and paste things because fields are different, but the concept is,
yeah,
how do you increase transparency? How do you consciously increase it, not just by one person, but a particular field coming together to say this is the direction we want to walk through. So it's really important. So everybody, please feel free to ask questions. I have one here that has been written on the chat. It says, thanking you for the beautiful lecture, and it's asking, can transparency be influenced by limitations of knowledge and facilities?
In which way? Yeah, well, I think if you don't know, if I interpret the question correctly now, if you don't know the limitation of your method, for example, you will not even know which parameters you need to be transparent about. So if you don't know that it's a risk of contamination, you will not look for it. And I think that's what the MySev guidelines and the EV track is trying to get people aware of these, because if people are aware, of course you want to do this, right? You wanna produce good, stable data, but if you're not aware of it, then of course you're in the dark. So yes, I would think that transparency will increase if we educate on limitations and increase the knowledge, for sure.
Yeah, thanks very much. Any more questions? I see so many thanks on the chat, which is quite good.
I mean, I hope I inspired you to think about your own fields. I know, as Bright said, you cannot copy paste EVTrack or something like that, but you can think about your specific field and what kind of problems do you have, and how can you address them? Can you work with other people to form like a similar group of people and attacking it in this kind of way? I think every field is benefiting from it, for sure.
Yeah, yeah. Cecilia and I had this course 2 years ago, as I told you earlier, and last year, yeah, that's last year. It was actually in November we had one. So in 2 years ago, one of the initiatives one of the groups prepared was to raise a campaign at the university, print T-shirt, print cups showing we want reproducibility. So the fact is that, I mean, this thing you showed somebody who started this EVTrack. So the movement can be started by one person. So these people we have in the course here, going back to your department, going back to your institute, going back to your centers, I think you have a motivation to begin to now have that level of self-criticism in what you do, to ask, how can we do things better for the sake of doing good science? Because eventually, if science is done the right way, it is a society that benefits. Every one of us benefits.
Yeah, and I think so. I know Anne Hendricks. She's a researcher like I from Belgium who started EVTrack, and since she's part of the society, it makes EVTrack not static. It was not made 8 years ago, and we have the same. When the field involved, they are adding that. So and making it a live, not document, but a live source that are listening to the community, and I think that's making it so nice. And I think the MISEV guidelines, I mean, we're already in 2021, maybe 2022 it will come a new one, because the field, as I said, is evolving so fast, and maybe the guidelines we had a few years ago are not so relevant anymore, or new things have come. So it's also nice to keep it alive, the process.
Mm-hmm. Someone is asking here whether it will be possible to get your presentation slides.
Yeah, sure. I think so. I will have a look at them, but I don't think it's any unpublished data in that case. I might just remove that, but otherwise I think that would be fine.
Yeah, when you're ready, just send to me, then I will circulate to them. Anyone else having any thoughts on this? If not, let's thank Cecilia. If you can give a hand of applause, please do so.
Sorry, Dr. Wright.
Who is speaking? Olanyika.
Yeah.
Okay, go
thank
ahead.
you, Dr. Cecilia, for that presentation. Now, yes, from my own field, biochemistry, molecular biology, which a bit relates to what we are doing, which is great. I really appreciate your presentation. Now, I want to ask, how do we deal with failed transparency? Now, in molecular biology, for example, like you rightly observed, somebody will publish, state the primers he used, state this. Now, you yourself, because you are interested in such a work, but using something else, you take that primer rather than going through the, try to troubleshoot in terms of primer. You take the published primer, you used it. Unfortunately, there are no bands. It's not amplifying. Nothing is coming up. You need to wonder, this primer was gotten from a highly reputable journal. I mean, I've lost money.
Yeah.
Not, you know what it means to all the collagen kits, all the rest, extract your DNA, only to discover when you are running your gel electrophoresis, no band, nothing is coming up. I mean, the same thing, toxicology. You try, somebody says so quantity of toxicants was administered, and this and this and this is what we observe, this and this. Now, you use that normally when you are also trying to do your own work. Okay, since this dose has caused this in so and so, definitely it will also cause this in this my animal model. You use that same toxicants, the same dose, only to discover nothing is happening. Not because maybe the person is reporting wrongly, but because there is no clear transparency, detailed step-to-step procedure. So how do you address that in like molecular biology and toxicology? Thank you.
Yeah, I mean, as I said, I don't think the majority of people are committing fraud. I think it's just the information is so little that we cannot repeat them in a good way. And as you say, it costs money and time for all of us. And of course, nobody wants to build a whole project on saying this did not work. We test something when it doesn't work. We order another antibody or another primer or something, and we just forget about that. We don't waste any more time and money. And in a sense, in that way, other people might try them as well. So it would be nice if there was a possibility to, I mean, maybe you can write like a small letter to the editor of that journal and highlight that I have tried this and I cannot repeat the data, or although you might not have the time to make a full paper out of it, so to say. I also think we should be wise when we reference paper. That's one of the tools we have as fellow researcher. I mean, I would never reference a paper that has 0 score in EV-Track, because it doesn't matter how cool even if they say they can cure cancer, I wouldn't reference this as, you know, an EV paper that have shown that. I would reference papers that I believe in, where I think they have been transparent enough. If I'm a reviewer, I always ask for this. If I'm a reviewer and I get an EV paper, I ask them to fill in EV-Track. I ask them for Western blots of non-EV proteins. I think that's what I can do as a fellow researcher on a lower level.
Thank you very much.
Okay, Olayinka, your question was answered. Does anyone have anything else to say? Otherwise, Cecilia, thanks very much. I appreciate this time you have given to us in this session.
Yeah, thanks for having me.
Yeah. Okay. Everyone will now will be expecting Go Chang, who will present for us next. He's not here yet, but if you have something to speak about, please just feel free. Go Chang has still 4 minutes, so you take his time. Well, you can stretch your legs. Let's come back in 4 minutes. Stretch your legs. Go Chang is here now. Go Chang, you're welcome.
Hi, Bright. Thank you.
Good morning.
Morning.
Yeah. Yeah, it's good to have your picture because they have seen me enough, so it's good that they will see you. while you present. But Cecilia just finished, and we are on just a short break waiting for you. In 3 minutes, everybody will be online. So you then I will introduce you.
Thanks.
Mm. Okay. I think we can kick off now. This is Go Chang. Go Chang is doing his PhD with me, and he's originally a physician, and then he's doing his PhD now in clinical epidemiology. So I will let him introduce himself properly and then kick off the presentation.
Okay. Thank you very much, Bright, for this very good opportunity. So I will share my screen. Sorry, can you hear me?
Yeah, we can hear you, Go Chang. Go ahead.
Okay, yes. Hi, everyone. My name is Guo Qiang, and I'm a PhD student from Bryce Group at Crafting Research Center. And today, the topic I would like to present is my PhD project, and it's about female sex hormones and health outcomes or asthma in women. And for today's presentation, I would like to talk about the two projects. Like one is the one that I'm currently doing, and the other one is the one that I completed. So For the first one, it's about endogenous and exogenous sex hormones and adult onset asthma in women. And the second is about menopausal hormone therapy and women's health. It's an umbrella review. And in the second project, we will talk more about how we implement the reproducibility project. So for the first, it's I would like to give a brief background information about female sex hormones. The female sex hormones mainly have like two types, estrogen and progesterone. And we can also divide it by its source into like endogenous and exogenous. For endogenous sex hormones, like such as we can measure that by the age of menarche or age at menopause. Like for example, the longer the women are in the reproductive phase, then the more exposed the women would probably be exposed to. And for other factors such as menstruation, pregnancy, etc., this key hormonal period or key event, then the women are also subject to like some fluctuation or increased levels of sex hormones. So this is endogenous sex hormones. And for exogenous sex hormones, there are like mainly two types. Like one is oral contraceptives, and the other is menopausal hormone therapy. And we can see they differ slightly from each other. And also the main difference is that like for oral contraceptives, it has higher dose. So it's used to reduce or suppress the hormone production. And for menopausal hormone therapy, it's usually used to supplement the hormones in addition to what we already have. So it usually has a lower dose. So for the first project, is about endogenous and exogenous sex hormones and adult onset asthma in women. Before we go like deeper into this project, I would like to also give a brief background about this field and why we are interested in answering this question. So as we can see from this study conducted in Finland, before around puberty, the prevalence of asthma is higher in boys than in girls. But after around puberty, and this prevalence is dramatically higher in females than in males. So there is a switch in the difference in the prevalence. And the same pattern was also observed in incidence. So before puberty, the incidence of asthma is higher in boys than in girls, but after that, the incidence is higher in women than in men. And this phenomenon was also in another cohort study conducted in US, and it found that by the age of 40, the adult onset asthma phenotype becomes the dominant type in women, which accounts for like more than 50%. And also in the VESAS cohort, which is based in Gothenburg in Sweden, and it found that the incidence rate of asthma in females is twice that in males. So given those observations, it seems like the sex or sex hormones may play a quite important role in the pathogenesis of asthma. So our group also conducted a systematic review to see the evidence base and what is current evidence around this topic and what we can do further to move this field forward. And based on the systematic review, and we found that the current evidence is quite limited, and it's restricted by like the amount of evidence available now, or the quality, the methodological quality of those studies. And here is one example about oral contraceptives and asthma. Based on the evidence from cohort studies, and the evidence, we can see that it is not very convincing, and we are not sure if what the effect of oral contraceptives would be in terms of asthma risk. And recently, our group also conducted a cohort study. We found oral contraceptives is associated with lower risk of asthma, and this association is duration dependent. And for menopausal hormone therapy, or we can also call it back then like hormone replacement therapy, or study, they are quite consistent, and almost all of them suggest that MHT is associated with higher risk of asthma. However, the recent study from our group also found different results. We found that menopausal hormone therapy is associated with lower risk of asthma. So we can say from the evidence we currently have, it's quite controversial about the role of sex hormones in the. So in our project, we try to investigate whether endogenous and exogenous sex hormones is associated with development of asthma, adult onset asthma in women, and also it may, if it would be associated with the manifestation of symptoms or clinical outcomes, and also the expression of markers of respiratory health and inflammation. And the study subjects we are going to use comes from the VESAS cohort, which, and briefly, there's like two studies. One is VESAS 1, and it included participants in 2008, and it invited 30,000 participants, and only like around 18,000 participants responded and agreed to participate. And this is VESAS 2, and it started in 2016, and it invited 1,000 participants, and around half of those participants responded. So for the project plan, what we are doing now is to imputing and coding the data, and we expect to start the analysis and get the results this year. And we also plan to implement reproducibility in this project, such as drafting and sharing the analysis plan and sharing the analysis code, etc. Yes, so this is the first project. And yeah, the second one is about menopausal hormone therapy and women's health. And it's an umbrella review of systematic reviews and meta-analysis of randomized control trials and observational studies. Yes, I'm going to introduce this project like in 3 aspects. One is about background, why we are interested in this specific research question, and what is the evidence currently available, and the methods, how we want to address this research question. And in this process, yes, again, there's some brief background information about why we wanted to do this.
Sorry, Gochang, just to say that
yes.
your line breaks, so it takes some seconds to come back, and then we don't get what you have said between that. So just to let you know.
Okay. Yes, sorry. Yeah. May I repeat what I've just said?
I think you can go ahead. So I don't know if there is an issue with the connection or something like that.
Okay. Yeah, so there are 4 stages of menopause. And we can say that from premenopause, the estrogen levels start to like fluctuate and decrease. And after reaching menopause, the estrogen levels finally reaches stable, but at a very low level. And during this menopausal transition, and many women may experience different kinds of symptoms. The well-known symptoms, for example, the hot flashes and the night sweats. And some women may also experience other symptoms like anxiety or bleeding or like incontinence, back pain. And during this menopausal transition, and some people also believe that this can cause also some other health problems. So many studies investigated the effects of menopause. And so they found that compared to late menopause, early menopause was associated with a few benefits, but also a lot of harms, such as increased bone fracture risk, coronary heart disease, mortality, depression, type 2 diabetes. So these menopausal symptoms and those health risks are thought to be mainly due to the fluctuation or reduction of sex hormones during the menopausal transition. Then it makes like quite a lot of sense to expect that if we supplement these hormones that we reaching menopause, it's quite likely that it could reduce those symptoms and also could prevent some diseases or reduce the risks. So there are many types of menopausal hormone therapy, and one is estrogen alone therapy, and another is estrogen plus progestin therapy. There are several other therapies, such as selective estrogen receptor modulators. But in our review, we are mainly interested in therapy and also estrogen plus progestin therapy, because those two are the classical types of hormone therapy. Around like 5 decades ago, hormone replacement therapy, back then it was called like that. So it was believed it could preserve the women's youthful appearance and keep her sexually attractive and preserve the strength of the bones. And so like back then, it was very popular and many women were using MHT. Around 2000, the largest trial to date, called WHI trial, published their data, and they found that MHT has some benefits, but it also has many like harmful effects, and some harmful effects were thought to be not to be related to MHT. For example, MHD was used to believed to be associated with lower risk of mortality, coronary heart disease, cardiovascular disease, but MHD was associated with increased risk of like stroke. So, and also it's increased risk with increased risk of breast cancer. So back then, the use of MHD dropped dramatically. And currently there are around 5 million current users in UK and the US based on the data available. And for the effects of MHT, yes, its effects are still quite controversial despite several decades of research. And for its benefits, we are quite confident to say that MHT could help alleviate vasomotor symptoms and could reduce the risk of fracture. But for many other outcomes, especially critical outcomes, such as coronary heart disease mortality, it's still controversial. And for its harms, like breast cancer, ovarian cancer, gallbladder disease, and asthma, as we discussed before, is still, the evidence is still controversial. So, recently there are several guidelines published by several leading menopause societies. And we found that in those guidelines, there's some inconsistency in some critical outcomes. And another issue we are a bit concerned about is those guidelines mainly incorporate systematic reviews as the key evidence support for their recommendations. So for a brief introduction about systematic reviews, systematic review is a tool to summarize the evidence surrounding one specific topic. And it's very useful if, for example, for one specific topic, usually we have more than one study. And it would be very useful if we summarize all these studies to assess the evidence base to say if the intervention or the exposure is actually has some effects on the outcome of interest. So this is systematic review, but it's very useful, but it's also associated with many shortcomings. One is that it's a short-sighted view of the evidence. Menopausal hormone therapy as an example, it has many effects, and usually one systematic review only summarizes the evidence for like one outcome or several outcomes. But for clinical decision making, we need to consider all the health outcomes, especially critical and important outcomes. So these systematic reviews, single systematic reviews cannot satisfy our needs for making our clinical decision. And we also found that for one specific topic, usually there's more than one systematic review available, which makes for the clinicians or researchers quite difficult to decide which one they should believe, especially when these systematic reviews make different conclusions. So it would be quite confusing. And the last one, and also very important one, is that empirical evidence. A lot of this evidence found that it's very easy to create a poor quality systematic review and meta-analysis. So to answer the research question, we in our umbrella review project, we try to what we umbrella review, what it does is to summarize all the systematic reviews related to this question, and we evaluate the methodological quality and credibility of these systematic reviews. And we would be able to get a comprehensive view of the evidence landscape to inform evidence-based guidelines and medical decision-making. So from this hierarchy of the evidence synthesis, we can see umbrella reviews stands at the top of the pyramid. And so basically, umbrella review is a high-level evidence synthesis of previously published systematic reviews and or meta-analysis. For example, if we are interested in the effects of intervention A, and then what we would do is to summarize all the systematic reviews which have looked at intervention A and any health outcome. By doing that, we would be able very quickly and also comprehensively to get like bird's eye view of the evidence, which can be used clinical making. So back to our project, our objective is to systematically summarize the benefits and harms of MHD use in women. And we try to provide currently the most comprehensive evidence to inform practice and policy. And also very importantly, to appraise the quality of those systematic reviews which have contributed to the current guideline development. There are around 7 steps involved in this review process. And the first is protocol. And the protocol was registered beforehand. And in this protocol, we specified the review question, and criteria, and finally a brief data analysis plan. And for the inclusion criteria, we are specifically interested in the effect of any menopausal hormone therapy compared to no use of menopausal hormone therapy on any health outcome or indicator. In any country. And the study design, the studies we want to include is the systematic reviews and or meta-analysis of randomized control trials and observational studies. And then we searched the database. We searched 12 databases. And we also, like in our paper, we also provided the search strategies for each database. And then we did the search study selection. So 2 team members independently screened these records and resolved by discussion. And finally, we included 121 outcomes. And those outcomes vary from different cancers to different cardiovascular disease and also to some surrogate outcomes. And each symbol represents this symbol represents like systematic review, which has looked at MHT and any health outcome. And then we did the data extraction and quality assessment. And for the data extraction and quality assessment, we, like 2 investigators also did this separately and compared with each other to ensure this consistency and accuracy of the data we would use. And also we noticed that based on a small sample of those systematic reviews included, we found that a poor data quality was quite common. So using the first 5 systematic reviews, we developed data extraction protocols to ensure that to ensure we get the data we want. And And for the quality assessment, we used the ARMSTAR2 tool to assess the quality of systematic reviews. And then finally, we did the analysis. And for the analysis, before we actually did the analysis, we drafted an analysis plan, which also went through several revisions. And finally, yes, we got the final version of this analysis protocol. And according to this analysis protocol, we wrote the analysis code. And for the analysis, we also considered like if we want to implement reproducibility for other results, then like software is also very important. So we used the open source software, which is the R software, so everyone can access that. And we also provided the analysis code, and with a very detailed description about each step from data importing to results output. And lastly, we shared the data sets we extracted, and along with the data set, along with the analysis code, and we shared it on the like OSF online repository, so that interested investigators would be able to access that if they want to like verify our results or reproduce our results. And then for the results, finally, yes, during this analysis, we try to provide these results as detailed as possible. And so we have a lot of online tables with very detailed data, the analysis results. Yes, and yes, that is how we implemented the reproducibility in our umbrella review project. And finally, I would like to talk a bit about why we were interested in this reproducibility. Yes, one advantage is that it can increase transparency. If the readers want to reproduce our results or evaluate the validity of our studies, they could go there and get the code, get the data sets, and the same results. And another advantage is that it could facilitate the peer review process and discussion with the reviewers and readers. If they have any questions, they can first like look at our analysis plan or our supplementary files. And if they don't agree, they could get back to us, and then we can discuss that. So they have enough details to comment on. And also, yes, I personally find it a very great opportunity to learn. For example, for the analysis plan, because I got to know that there are so many methods, and each method has their own advantages and disadvantages, and so which makes it difficult to decide which method we're going to use. So by implementing this reproducibility, we would be able to like have the opportunity to think it through, and which method is most suitable for our project, and then also provide the justification for that. Yes, then that is all for my presentation, and thank you very much for your time and attention. Thanks.
Okay, thanks very much, Go Chang, for sharing with us what you have done in your project. Does anyone have questions for Gochang? Questions or comments? Does anyone? The idea here was to bring people from different research fields. What Cecilia presented in the morning, you could see, and then what Gochang has shown here as well.
Sorry. Yeah, the internet just got disconnected.
Yours, eh?
Yes.
Tekla, do you want to say something?
Yes, I have a question.
Yeah, please go ahead.
Okay, thank you, Goshen, for your presentation. I have really learned a lot. But I have a question. I want to know if MTH extend the onset of menopause.
But I didn't get that.
Sorry,
Tekla, is it possible to write it? And because you have background noise, so we can't hear you clearly.
Okay, let me type it here.
Yeah, so Gochang, she will write it on the chat, then I will let you know what she is asking for. Anyone else? Questions, comments? Okay, Tekla is asking, does menopausal hormone therapy extend the onset of menopause?
Okay. I'm not sure because usually when people they are like reaching menopause and then they have symptoms, then they start to have menopausal hormone therapy. So it's like they don't use it before they reach menopause. It's like after reaching menopause, they have some symptoms, they start to use it. So yeah, I haven't read any papers regarding the effect of menopausal hormone therapy before reaching menopause and how it affects the onset of menopause.
Okay, thank you for that. There is another question here. In your experience, did you find any challenges in carrying out this reproducibility study?
Yes, actually, I found it quite challenging because like one main reason is the time, and because in the beginning I was not very familiar with reproducibility and how much details I should like record or put it in document when doing the research project. So the main concern or the main challenge I was having is the time. And also, if another thing is if I want to like do this implement reproducibility, and how much knowledge I should have. And like for example, there are so many methods out there, and if we decide to use one method or several of them, what will be the justification for that? Because each method they may produce different results, and the different results may be interpreted differently. So yeah, I think that is another quite challenging part. But the good thing is that when doing in the umbrella review project, we also have the help from a statistician who has expertise in this field. So then other than that, yes. But then if we get familiar with this reproducibility, then later for later projects, then it would make it quite easier to do this, and we can like benefit more and with less time. Yeah, thanks.
Okay. Yeah. Any other thing? Any other question? Okay, I have one here. It said, could you kindly give more light? Give more light on the benefits and harms of MHT. Not too clear, for instance, how mortality or additional risk of cancer could be beneficial. So can you throw more light on the harms and benefits? Go Chang, you have submitted your paper, so it should be with a journal. Do you want to give us some highlight of your results so that those who are hungry to know what you have found? John is a man. He's asking of MHT. So well, it's good for knowledge sake. So Go Chang, up to you.
Yes. Yes, I would love to. Yeah, so what we, the general idea with from this project we got is that from those guidelines, many of them are like saying that MHT, the benefits of MHT may be more likely to outweigh the harms in young women. So they times, and they say MHT can even like reduce the risk of all-cause mortality and coronary heart disease in young women, but not in older women. Though MHT is in general associated with many other harmful effects, but because of these benefits in young women, and these are also critical outcomes. So they, it's kind of the balance is leaning towards the benefits this side for young women. But from our review of those evidence, we found that the evidence supporting this timing hypothesis, which is like on coronary heart disease or all-cause mortality in young women, is quite tentative. And so yeah, the general like impression from that is maybe MHT, like the balance in young women is quite similar to older women. And so then, yes, we also like suggest in our manuscript, maybe current guidelines may need to like re-examine, like experts in this field may need to re-examine these guidelines to say if we need to like be quite positive about MHT use in young women. Yes, I hope that is clear.
Yeah. Any other? Which part of the world did the papers you reviewed, I think, come from? And the person is curious about papers from Africa, studies from
Yes,
Africa.
yes. So for the embryology project, we include reviews.
No, we didn't
Review.
get that, Go Chang. Your line broke.
Sorry. So we mainly included systematic reviews in our project. And for those systematic reviews, they included many individual studies from different parts of the world. But we were not able to like identify which study comes from which country. But I feel that it's quite likely like the evidence base or the individual studies come from many countries or most countries, including like Africa, Europe, and US, and many other countries.
Okay. Any other thing? Any other person with questions? Okay. If not, Gochang, thanks very much for sharing your time with us and giving us a highlight of what you're doing and how you're thinking about these issues of reproducibility in your research. I think the goal, as we have re-emphasized, is to ensure that we have transparency, ensure that we increase the benefit science has for the population, for the society, and to eliminate things that will make, once the results are published, the society has doubt because they think that the researchers may not be doing things in the right way, to avoid fraud and things like that. So this is an example of a field, how somebody is implementing it, and I think we can learn from this in going forward in our own areas of interest. Thank you again, Gochang. Have a good day. Please, if you are able to give him a round of applause, please do so.
Okay. Thank you very much, Bright, and thank you very much, everyone, for this opportunity.
Great. Okay. For everyone, perhaps you can take a 10 minutes break, and when you come back, I will send you back to your groups so that you can continue discussion of your project. We still have more than 30 minutes before the lunch break. So please take a 10 minutes break and let's come back 25 past the hour.
Okay, Dr. Bright.
Yes, Felix.
So I don't know if it would be possible to have the protocol before our break.
The ones you want me to, yeah, I will do that. I will do that for sure.
Thank you.
Yeah. Okay. So I will send you to your groups now. And the plan for today is that before the launch, you can continue your work. I have received from group 3, I have received their plan for the Thursday work, which is fantastic. Really, I went through it quickly, but it's fantastic, which means that you understood what you're supposed to do. So thank you for that. So I will await from the other groups. And so please, if you have not formulated your questionnaire, do so. For group 3, when we return from the launch, you have to extract your questionnaire, and if you can show your slides, we can do that. And so the other groups, if you are able to finalize your questionnaire by the time we return from the launch, that would be great. But if not, we'll see how we'll do that. So the plan is that once we return from the launch, we will work to harmonize all the questions and then develop the questionnaire and put on perhaps Google Forms so that we can all fill it. And so after that, the remaining part of today, you will work on your project, do the analysis plan and things like that. And then tomorrow morning we'll come back and we'll use the morning hours of tomorrow to finalize the project for tomorrow, because we'll be presenting it once we return from lunch tomorrow. So after that, after the presentation, then you have time to continue with your project for Friday, and then we'll continue with that on Friday as well. Okay, so I will send it to your groups now so that you can continue on that. And as usual, I will just mention your name and you let me know which group you are. And sorry if I keep repeating this because I don't have them. Yeah, I have somebody raising the hand. Is that the case? Somebody wants to speak? Ask question? Are they with me? What group are you?
Group 4.
Group 4, yeah. Amanda is group 1. Beatrice Karume. Beatrice Karume. Claire. Claire, what group are you? Clotilde.
Hi, Clotilde, group 1.
Group 1, Clotilde.
Helen, group 4.
Claire, you said group 4?
Yes.
Okay, Claire, group 4. Defina?
Group 4.
Group 4. Most of you are group 4. Tabitha? Is Tabitha there? Tabitha? It's group 3? Did I hear group 3? Eman is group 8.
Yes, thank you.
And Felix is group 2 you are?
Felix is group 7.
Group 7, sorry. Ako is group 8. Ngozi is group 3? Ngozi, what group are you? Ngozi is not there. Valerie?
She's in group 3. She's in group 3.
She's in group 3.
Sure. Yeah.
Valerie?
3.
Group 3. I'm trying to unmute.
Great. Benga Asaolu. Valerie, you disappeared yesterday. We didn't see you when we were talking about you. Benga Asaolu. Godfrey.
Group 2.
Group 2. Job is group 3, eh? Job there. Jocelyn?
Yes.
Oh,
yes.
great. Jocelyn is group 1, I think. John is group 2.
Yes, sir.
Johnston.
Group 3.
Group 3. Kellen. Kellen.
Group 4.
Group 4. Marion.
Group 7.
Group 7. Nelson. Nelson group 2. Olayinka. Olayinka Oko.
Olayinka is in group 4. Group 4.
Group 4. And Ruby Kodom. Group B.
7. Group 7.
Okay. And
I'm in group 5, see, please.
Thank you.
nursing,
I'm in group 5. Dr. Bright.
Yeah, nursing,
No, Benga. I'm in group 5.
Benga. Benga, do you have somebody in group 5 with you?
I was in group 5 with Emman, but I wasn't around yesterday.
Okay, so join them in group 8 because we have moved Emman since you didn't want to come yesterday. So now I send it to group 8.
Group 8, all right, sir. Okay.
Yeah. Lee is group 1. Tekla Akukwe. Tekla, are you there? Theo.
Oh.
Theo, what group are you?
Group 2.
No, I didn't get that.
I'm in group. Said it's also you. I'm in group 2.
Group 2. And
Oh, yeah.
Zelalem. Zelalem. I think most of you already have your group, so just go to your group.
Hello, Dr. Brett.
Over here, it's pretty
Yeah,
hard.
who's speaking?
Yeah. So please, I just wanted to clarify something. The way the project looks, it seems it's a quantitative project, not a review. Please, could you clarify?
No, it's not a review. It's not a review. It's a project. You go to the field to collect data and analyze it.
All right. Yeah. Thank you.
Yeah.
Yeah, sure.
Okay. Let me see these ones that have not. Nursing is group 2, so I send him to group 2. Elizabeth, Tabitha is group 3. Beatrice, Karume, Zilalem. Group 7. Tekla Kukwe. Group 7. Beatrice Karume, I don't know. Okay. Group number 1. Are you there?
Yes, please, we are.
Yes. So the lunch break starts at 12 noon my time. So just at the hour, about 20 minutes from now, and then we will resume again at 1 p.m. in the whole group.
Okay.
Yeah.
Yes,
All the
sir.
best.
Okay, sure. Thank you.
Okay. Group 2.
Yeah.
So the lunch breaks is in 20 minutes time, and then we'll resume at 1 p.m. So 1 hour after that, we'll resume in the whole group.
Yeah.
Okay.
Okay, Prof.
Thank you. See you later.
Right. Just a clarification on some key aspect of our assignment and also reproducibility. I think I was not there for the first day. We had the internet challenge and all this. I've been reading, and then now the two presentation from your colleagues there have tried to strengthen, and our colleague also seem to be in the same dilemma, how to conceptualize this, because for my own understanding, which I'm getting from all this presentation, is while you are doing your study, there is a need for a strong consideration of transparency, which you keep emphasizing, which I fully agree, which I fully believe. And that should take precedence right from the process when you are conceptualizing your topic, defining the protocol, going through the data analysis and the whole process. And then somebody now is asking, he doesn't feel that process, he doesn't see it. How can that be broken down? And this will help us in putting our also topic for tomorrow.
Yeah, just to give example, group 3 has done exactly what I expect them, which is fantastic work. They've actually proposed hypothesis based on the research question I've given them. They have proposed a hypothesis they want to test. And then they have proposed different objectives they want to evaluate that will lead them to addressing the hypothesis. And then they have indicated the method they will use to collect the data. And of course, I said that the participants in this study will be those who are participating in this course. And so they have also developed a questionnaire of they will use to collect the data, and they have developed questionnaire questions on demographics, for instance, what is your gender, your age, and they have developed questionnaire around answering each of those objectives, answering question from the participants on your awareness, your perception, your practice of reproducibility before coming to this course. So I think those are the things you need to do. One aspect of reproducibility we have emphasized that you need to consider is development of a protocol before you go to the field to collect the data. So the protocol should be evident that you have thought about this and you have mapped out what you would want to do, what you want to get, your questions, the variables, the data you want to collect. And then once you have collected the data, you need also to plan the analysis as well, dealing with the data. So you need another protocol as well at that stage to indicate what you want to do with the data, how you want to, for instance, if age was collected as a continuous variable, for instance, you want to categorize it. How are you going to categorize it? What program are you going to use? How will you develop your coding of the variable? And once you have started your analysis, you need to also ensure that you have a syntax, and all this should be transparently done and intelligible so that once you are not there, somebody else can work on that and use your protocol and use the issues of reproducibility you have considered to run that study and derive the same results you have derived. Did I help you?
Yeah. Other colleagues, Jordan, Theo.
Yes, I am there.
Oh, yes, it does.
Okay.
It's helpful.
Thank you.
No problem.
Thank you, Prof.
See you later. Hello, group 3.
Yeah. Yes,
Yeah.
Dr. Bright. You
So
can
this is
hear
the
you.
group that did a fantastic work. So the break starts in 14 minutes time, and that is the lunch break. And then we will come back 1 hour after that to continue what we started together. So just for you to know. Do you have any question?
Yes, Dr. Bright. Yeah, so we're just thinking in terms of looking at the demographics. So maybe do you have any feedback already from what was shared already?
Well, I plan to give the feedback when we meet together in the group once I have what others have done as well.
Okay. Then I think, yeah. Then I think that's fine.
All right.
Just
Okay.
Thank you very much, and see you later.
Okay.
Hello, group 4.
Yes, sir. Good afternoon, sir.
Good afternoon. So just to let you know that the break time is in 12 minutes time. That is the lunch break. And then we'll have 1 hour for that and come back to the main group after 1 hour on after the lunch. And then we'll continue from there.
Okay, sir. Then, sir, excuse me, sir.
Yes.
We have a question. Can we share our question with you?
Yeah, you can send me email.
Okay, of the questions alone or what we have done so far.
Yeah, if you want to send me everything, no problem. If you want to send me a question, no problem. If you want to keep it until we meet after lunch, that is also fine.
Okay. Okay, I will agree with our group member, so we know what to do. I would discuss with our group member. We know what to do. But we have some questions that we would want to share. Yes, sir.
All right. So
thank you, sir.
I'll take care.
Yeah.
Okay. The group, this group 7, eh?
Yes, sir.
Yeah. So just to let you know that the lunch break starts in 11 minutes. So you play the game the way you want it. We are resuming 1 hour after the break, after the lunch. I mean 1 p.m. My own time, so that you know, and if you have developed your question before then, that would be fantastic, so that we can take it forward from there. So if you plan to eat, then you eat during the lunch break, because after that we'll come back to the main group.
Okay. Thank you, Dr. Bright.
No problem. Take care. See you later.
Okay.
Four instead of 6.
Hello,
This,
group 8.
This is Professor Bright.
Group 8.
You're welcome,
Yeah.
sir.
Yeah, just to let you know that the lunch break is in 9 minutes, but you use your time the way you want it. But one thing you have to know is that after the lunch, that is 1 hour lunch, we are meeting in the main group to continue the discussion.
Okay, sir.
Okay. See you later.
See you. Thank you.